Hi everyone. Chapter 7 reviews physical structure of chromosome, and this chapter covers genome packaging, chromatin structure, and metaphor chromosome feature. We will also review the organization of genes and other genetic components in eukaryotic nuclear chromosomes. As you already know, nuclear genome has enormous size. This portion of the genome is split into a set of linear DNA called chromosome. Since the chromosome is really long, in order to pack everything into small pieces and place in the nucleus, there gotta be the process of genome condensation is required. First, we will review the first observation of genome structure. Shown here is electron microscopy feature of the nuclear chromosome after they did lysis of the chromosome by detergent, and then they took a picture of the internal structure by electron microscopy. As you can see here, it looks like a bead on a string structure, as they named. This is association of DNA with the binding protein, but when they first discovered this, they wasn't sure what are the components of this structure. Now we know this bead structure are composed with histone molecules of their polymer binding. So the assay that they discover the bead on a string structure and see what the components is, they used a nuclear protection analysis. This study is commonly used to determine the binding location of the protein. The basic principle of nuclear protection assay is that when the nuclease digests the DNA, if there is any protein binding to the DNA, the location is not digested by the nuclease because the already bound protein protect those area from the nuclease digestion. So first they purify the chromatin structure and they digest this structure with nuclease. So they use the two different time points. One is the limited digestion and the next make a full digestion. The reason why they do limited digestion is to see the intervals between those nucleotides when it's wrapped around by the histone protein. What they discovered, so you digest a nuclear DNA complex in various time points, and then they run on a gel. When you run on a gel, if the DNA is less digested, the size of this fragment is much larger than fully digested components. And if you can measure the DNA size of the area that's wrapped around by the histone protein, then you can determine the length of the DNA that's associated with the protein. So after they do the electrophoresis, what they found was that there are 400 base pair, 200 base pair, and 146 base pair with the full digestion, which indicates from the partial digestion, you result in 200 base pair as a monomer, you result in 400 base pair as a dimer. So which means the area including where the histone molecule associated plus some of those side residues are about 200 base pair. The area where it's only wrapped around by the histone protein is around 146 base pair. So, from this assay, they figure out that the DNA is associated with the histone molecule at every 200 base pair, and the core region that's wrapped by the histone molecule is about 150 base pairs. And later on, they isolate those proteins to see what type of proteins are associated with the DNA, and found there are 4 different core proteins forms octamer structure to form the core wrapping protein, and then there are additional histone molecules formed in between those 2 histone octamer to stabilize the DNA bead on a string structure. So the core octamer is formed by H2A, H2B, H3, and H4. Those are stacked into two layers to form 8 molecules form one histone complex. The key terminology here is that when the histone complex is associated with the DNA, we call those individual strings as nucleosome. Then what determines those 50 base pair interval between the adjacent nucleosome? There are other protein molecules called linker histone. The linker histones H1A, 2E, H10, H1T, and H15 basically form a bridge in between the nucleosomes to stabilize the nucleosome structure. So the entire complex, including the histone octamer association with the DNA and the linker histone, we call chromatosome. So two terminology here that you need to remember is one, nucleosomes, which are the DNA complex with core histone molecules of H2A, H2B, H3, and H4, and the chromatosome, which is more stabilized structure including linker histones. Okay, then the bead on a string structure is not the actual chromosome status that's found in the cells. The bead on a string structure when they first discovered the nucleosome, but the actual process was doing a lysis with a detergent which denaturate the DNA and protein association. So later on they did more in-depth electron microscopy and they figured the actual structure DNA condensation is more thicker than a bead on a string structure. We call it as a 30 nanometer chromatin. 30 nanometer chromatin fiber is formed by supercoiling of the bead on a string structure. So individual loop indicates the bead on a string structure formed by histone association. It goes through the supercoiling, form much thicker structure. Still, it's not clear how the supercoiling formed, but there are two models suggested. One is solenoid model, the other one is helical ribbon model, but still need to be determined for understanding more precise structure. The 30 nanometer chromatin fiber is the major type during the cell cycle, as most of the cells are in the interphase status. You often see 30 nanometer chromatin fiber, and you can only see the chromosome structure during the mitosis. This is a more detailed image of the histone association with the DNA, shown here in turquoise and the yellow. These are the DNA double helix structure. Blue, green, yellow, and red colors are 4 different histone molecules indicated by different color. And you will see there are some linear structures stick out. Those are the histone tails. So we will review this later, that the histone tails are important in managing the nucleosome structure and change the condensation status. Because depends on what type of modification occur on the histone tails, it can make much more tighter binding and it can form much looser binding. So as a mathematical equation to determine the size and the density of the DNA structure. So as a quick calculation, the 10 base pair DNA is 2.4 nanometer in width and 3.4 nanometer of the 10 DNA stacks. And when it forms nucleosome structure, there is at least 7-fold condensation. So it will form 10 nanometer bead on a string structure, and when it forms 30 nanometer fiber structure, which is normally found in the native states of the cell, and form 100 nanometer loop structure, which is much more condensed structure, mostly in heterochromatin structure, and later go much further in the chromosome condensation, which is about 10,000 fold increase. So from the 3.4 nanometer by 10 base pair, and that's actually condensed into 1 micrometer size or smaller. So let's look into the typical structure of the metaphase chromosome. I mean, this is very familiar to everyone who take this class. So metaphase chromosome has a key critical components, two arms, which we call chromatid, and centromere, where it's associated microtubules to pull those nucleosomes by the end of mitosis, and telomere structure, which is the structure to protect the terminal end of linear DNA. If there's no telomere structure, then the nuclease can bind to the terminal end, can actually digest the DNA. So then how the name chromosome came with? Before the electron microscopy was evolved, you can only determine the intracellular structure by staining or observation under other techniques. The name chromosome came from its nature, that those structures are stained with a very unique pattern, and as it's stained by staining reagents, named after the Latin root chromo means the color. In somatic cells, there are two copies of chromosome of each chromosome found, and centromere, chromatid, telomere are the key term for indicating each form of the structure of the chromosome, as you already know. These are the actual staining pattern of each chromosome that we call karyogram. As it have a very distinctive banding pattern based on different staining techniques. When they first discovered this, they expect these locations are associated with gene expression or different composition of genetic elements. But later it's discovered that there is no clear correlation between the gene location and functionality of the each genetic components. So still it's not clear why it's stained in a different pattern, but there are several studies pointed that those area that's either AT rich or GC rich are stained in a different patterns by different staining agent. Giemsa versus quinacrine, they stained in different location, darker than other location. So mostly the chromosomes are the similar feature that it has two chromatid arms, centromere and telomere at each end, and has a relatively similar size. But among many eukaryotic systems, there are some unusual features of the chromosome found. One is mini chromosomes, which has a much shorter size than other chromosomes, but it's highly enriched with active genes. Macrochromosomes are much larger, but relatively low density of the genes are located on macrochromosome. B chromosomes are additional chromosomes on top of the normal numbers of chromosomes found in each system, and these B chromosomes are commonly found on plants. Holocentric chromosomes are very interesting structure with a multiple centromeres spread along their length. So let's look into the actual structural feature of centromere and telomere, because centromere and telomere has quite unique structure that you can't really find in the chromatid arms, and they govern specialized function for DNA replication and segregation. So the centromere structure has been extensively studied in yeast system, but still the study is going on for eukaryotic mammalian centromeres, because the structure of the mammalian centromere is a lot complex than the yeast centromere. So in this lecture, we will mainly focus on the centromere structure of yeast system, and we will compare and contrast the components with the mammalian system. So you will only need to focus on the centromere of yeast chromosome. So the centromere structure is composed with three distinctive domains, CDE1, CDE2, and CDE3, has different nucleotide base pair compositions, And those individual locations are recognized by specific binding proteins. So for example, CD1 is recognized by the binding protein called CBF1. CD3 is recognized by CBF3. And the middle region, CDE2, is recognized by the protein complex. When look at the proteins binding to the centromere structure, that they found that they have specific binding affinity to the DNA. For example, CES4 has a similar structure like a histone 3. CBF1 and CBF3 bind to at least 20 more other proteins to form kinetochore. So on this core region of the centromere, binding of CES4 and MIF2, binding of CBS1 and 3 form the core elements of the centromere, and additional 20 or even more proteins are associated to form critical kinetochore required for DNA segregation. So kinetochore is the location where the microtubule binds and pull the chromosomes by the end of anaphase of mitosis to separate duplicated chromosome. The centromere of the plants and animals are a lot more complex. So the protein complex called CENT-PA is highly similar to H3, which has the conserved domain to CBF1 and 3. The interesting feature about the plant and animals centromere is that the centromere region has high numbers of repetitive sequences. In particular, the repetitive unit of the human chromosome is about 171 base pairs, and at least you can see 1,000 copies of 171 base pair repeats, which is around like 1 megabase total size of the centromere region. And when there are highly repetitive sequences are found on the chromosome, and those region usually does not contain many genes. In the centromere region, there are not many genes are found, and those centromere seems like it's only govern the function of the physical segregation of the chromosome. Telomere also is formed by multiple repetitive sequences which are preserved after each DNA replication. Telomere is also required to form distinctive structure of the terminal end to protect the linear chromosome from nuclease digestion. At least 100 copies of the repetitive sequence, as shown here, the repetitive sequence unit is TTA, GGG. At least 100 copies are lined up, and those length of the repetitive units are maintained by a special enzyme called telomerase. Telomerase functions with two major proteins, one TRF1 and the other one TRF2. TRF1 is the protein regulating the length, so this is actual polymerase extend the telomere structure. TRF2 is the protein required for the terminal structure maintenance.